This video is going to focus on metabolism. And one of the things that I want you to be able to do um, early on is to be able to distinguish metabolism from cellular respiration as well as catabolism. They're all interrelated, but I want you to be able to distinguish among those. I'm also then going to look at metabolism of carbohydrates, metabolism of lipids, and then metabolism of proteins. And in doing so, especially with the carbohydrates, these terms oftentimes get mixed up for students. So glycogenesis, glycogenolysis, and gluconeogenesis. So what do those all mean? And under what circumstances would each of those occur? Similarly, to do the same with lipid metabolism, so we'll look at lipolysis, lipogenesis, and then ketogenesis. And so being able to distinguish when each of those would occur. And those are going to be some of my key goals for you all with this video. So first up, cellular metabolism. And so with metabolism, what it really is it's the sum total of both catabolic processes where we're breaking things down, as well as anabolic processes. So the sum total of all of those chemical processes is your overall cellular metabolism. So I love this graphic because you're seeing a little bit of digestion come in here as well. So you see diffusion or transport of your amino acids, lipids, and simple sugars, right? We know that this would occur in the small intestine, that we bring in those macromolecules there, either in the lacteal or through the capillaries. And then so what you're looking at from here is what happens to that nutrient pool, right? So with catabolism, this includes that process of cellular respiration, which I'll try to define clearly in a moment here. But catabolism is our chance to try to break down those macronutrients and to capture that energy as much as possible in the form of ATP. Now, you'll see that a lot of that, as efficient as the human body is, we still end up losing a lot of that energy in the form of heat, just given off from our body. The energy that we are able to capture in the form of ATP then goes to drive other things, right? So other cellular functions, mitosis, transport, active transport, right? That requires ATP, movement, muscle contraction, all of that. And then with anabolism, right? We also need ATP to drive this building process. But this is still part of the overall cellular metabolism. So this could be taking amino acids and building them up into proteins or collagen or skin or muscle that we might need. You see the synthesis of other organic molecules. So if we have too much glucose and we're building that up into the form of glycogen for storage, that would also fall under this process of anabolism. Let me build a little bit more on cellular respiration. So in previous classes, you have learned this, right? The mitochondria is really the mover and shaker here. You have glycolysis occurring in the cytoplasm, but then the rest, the Krebs cycle, the electron transport chain, that's all going to occur in the mitochondria. What you don't see in this picture, which you probably didn't study when you first learned cell respiration, is where fats and where proteins come into play with cellular respiration. And so to define this, again, this is going to be a group of catabolic, so breaking down those food fuels, breaking down glucose in this case, breaking down triglycerides, or breaking down amino acids as best we can to trap that energy in the form of ATP. So that is very much the ultimate goal of cell respiration, is trapping chemical energy in the form of ATP so that we can then use that to drive work. I think this is a useful graphic from your textbook, but I like that it does start off with this process of digestion, right? So this was the whole last chapter that we were looking at. So you can see proteins, breaking them down to those monomers, carbohydrates, breaking them down to glucose and other monosaccharides. And then our triglycerides, right, their monomers are both the glycerol as well as the fatty acid chains coming off of it. So that's the digestion piece, right? And then at that point, again, we bring things through the membrane of the small intestine, and then metabolism can start. So what you're seeing in this figure is all Anabolic processes are shown with the purple arrows. So we're going to see that with proteins, we take most of those amino acids, and rather than breaking them down for energy, we don't like to use proteins for energy. So rather than doing that, most of those amino acids will be anabolically processed to make proteins, right? To make the countless proteins that our body depends on to function. With carbohydrates, we know that these are an excellent source of fuel, as are fats. So we're going to do a lot more catabolism here, the blue arrows, where we're breaking down sugars from glucose to pyruvic acid. This should bring back some memories, acetyl-CoA. With fats, right, we're going to be breaking down glycerol. We're going to be breaking down the fatty acids. And then last, the rest of this is going to happen in the mitochondria, right, where we go to the Krebs cycle. And this is where we really get, where we really have the ability to maximize ATP production, is when we get to that electron transport chain and oxidative phosphorylation. Now, the details of these processes isn't my focus in this video. I really want to look more upstream of this. So how do we get from here to here? How do we break down these fats? That's what I'm going to be focusing in on more in this video. And we'll get to bring in some nutrition as well. So let's look at some of these G words. And I threw in this figure to try to tap into some prior knowledge here. So we saw when we looked at the endocrine system that when blood sugar is too high, so here we are, blood glucose levels have risen, we know that insulin is released from the pancreas. And the reason that's so critical is because it plays this role in metabolism. So insulin allows for our hepatocytes in our liver and other cells, namely in the skeletal muscle, to be able to take that extra glucose and to be able to store it in the form of glycogen. So we are making glycogen. So if you were to put a name behind that, let's try glycogenesis. So genesis means the creation of, and we are saying we are creating glycogen. So you really just have to break down these words, and they will define for you what they do. So as I said, glycogenesis is going to occur when the glucose supply is larger than the demand. So when blood glucose levels have risen, the cells can only store so much ATP, and we know that we don't waste glucose, right? Glucose is so critical for the function of the central nervous system that when we have too much of it, we don't excrete it in our urine. Our kidneys don't process that out. Instead, we store that in the form of glycogen, again, in the liver, skeletal muscle, the primary places. So that's glycogenesis. Now, glycogenolysis, we have also seen, I just didn't define it as such when we were looking in the endocrine chapter. So that's what you see down here. I've added the term. And so if blood glucose levels fall, it could simply be skipping breakfast in the morning, or even the overnight fast, that 8 hours ideally that we've had to sleep. To some extent, our blood glucose levels will fall there. Our pancreas will release glucagon. And it's going to do just the opposite, right? So the glycogen will be broken down, will be lysed, and we'll get glucose released, and it's released into the bloodstream. So it brings blood glucose back up. So we are saying glycogen, and then we've seen this root word a lot, lysis, right? It means to split. So we are splitting glycogen. Right? Makes sense. And we are only going to do so in situations where we maybe are a little bit fasted, or we are just generally hypoglycemic, so where our blood sugar has fallen. Last up is gluconeogenesis. We also saw this in the last chapter. If you recall, there was a hormone that's involved in the stress response that triggers gluconeogenesis. So it was cortisol. And so gluco, so sugar, neo, meaning new, and then the creation again. So we are saying it's the creation of new sugar. So this is an unusual one where glucose is actually made from amino acids, from proteins, and from glycerol, which comes from our triglycerides. So notice that I don't have fatty acids here. When you break down your triglycerides, you get the glycerol backbone, and then you get your fatty acid chains. So the fatty acid chains cannot be used to make sugar, but the glycerol backbone can. And it may surprise you, but you actually get more sugars made from the amino acids than you do from the glycerol. And so a case where you would have gluconeogenesis occurring would also be if you are fasted or if you are not taking in many carbohydrates. So with the ketogenic diet or just a really, really low carb diet, your body may very well be doing this, right? When we've gone through our glycogen stores and we can't make enough glucose, because glucose is so critical to our central nervous system, we find other mechanisms of making it. So what I was going to drive at is that with a very low carbohydrate diet, to some extent, we do actually break down lean muscle to get these amino acids to be able to provide our bodies with glucose. So want to clear up a common misconception here. So pause for a second and see if you can answer this true or false question. All right, so when at rest, our bodies are predominantly using carbohydrates for fuel. And that's false. So fat is the preferred fuel of our liver, of our cardiac muscle, and our resting skeletal muscle. So this gentleman here is actually in the fat burning zone. He's burning more fat than he is carbohydrates for fuel. And that's because fats are such a good source of energy. Recall that we get 9 calories per gram of fat that's used for fuel, compared to about half of that for every gram of carbohydrates that you use for fuel. So when the body can use fat for energy, it much prefers that. So let's look at some of the fundamentals of lipid metabolism. So this looks busy over here, but all that I'm showing is this is your standard triglyceride. So tri meaning 3, that's referring to the 3 fatty acid chains. It's basically a hydrocarbon. You've got a carbon backbone with all these hydrogens coming off of it. And then you have glycerol, and this is very much like a carbohydrate, which is why it can be used for that process of gluconeogenesis to make new glucose. And so when we split triglycerides, when we are catabolizing triglycerides, hydrolysis, we are moving in this direction, and you can see that we split to the glycerol and the fatty acids. We can flip that as well to make our triglycerides. So lipolysis is the direction going from the bottom to the top here. So enzymes, so we've seen these lipases now, right? We know that we have lipase, we've got lingual lipase, and we also have pancreatic lipase that are used to split these triglycerides. Lipogenesis, monomers join, and they are stored as fat in the body. And so if we consume excess fat, if we consume excess carbohydrates, and also if we consume just excess proteins, what we will do is we will store that in the form of triglycerides. So one thing that's a little different about triglyceride metabolism is the monomers are oxidized separately. Oxidized, if you think back to previous classes, so Leo goes Gur, right? So lose an electron, oxidized. So catabolic processes, right? We're breaking down, we are releasing electrons. Those are oxidative processes. So you can translate this in your head to say that monomers are broken down separately. Okay, so here's our lipase, here's our lipid from our diet, here's our lipase from our GI tract, or coming in from the pancreas, coming in from the mouth. Glycerol, fatty acids. The glycerol is treated like a carbohydrate because it basically is at that point. So it gets to go through glycolysis. It gets to be converted to pyruvic acid. It jumps into acetyl-CoA. This is going to become a really important intermediate. So bear this acetyl-CoA in mind. And then from there, we go into the Krebs cycle and we're golden. We get to go to the electron transport chain. We get all that ATP. So I'm not even showing the rest of this graphic. Now, on the side of the fatty acids, we go through a process called beta oxidation. And all I'm going to say here, well, there's actually a lot to say, but I'm going to try to simplify it. So I want you to know this term, beta oxidation, and I want you to appreciate that it's what fatty acids undergo in order to get acetyl-CoA. So if you recall, this is a kind of a side note, but CO2 is given off here. So this step from pyruvic acid to acetyl-CoA is not reversible. And so the fatty acids, when they are broken down to acetyl-CoA, they can dive into the Krebs cycle, but you cannot go backwards. You cannot go backwards and get carbohydrates or glucose from the fatty acids that are broken down. They can't go from acetyl-CoA to pyruvic acid. You can't just capture that CO2 like a plant can to get back to pyruvic acid. We can't do that. So that's why, thinking to gluconeogenesis, I emphasize that you can get glucose from this glycerol because it can be converted to pyruvic acid and you can go backwards, but you cannot get glycerol from the fatty acids. They are too far downstream. You can get energy. You can get lots of energy from this, which is awesome. We need that for just ATP, but you cannot get sugars. You cannot get glucose from the broken down fatty acids. Conversion of excess carbs to stored fat. So it is the case that if we consume too many carbohydrates, we will store that as fat. So in the 1990s, early 2000s even, right, these low-fat recipes were all the rage. And we were forgetting some fundamental biology with some of these dietary trends where simply, eating less fat does not translate to less fat on the body. Because if you eat less fat but are eating lots of carbs, then it doesn't matter. Our body can easily convert that over. So dietary glycerol and fatty acids not needed for energy in that moment are both converted to triglycerides via this process of lipogenesis, and they're stored in adipose tissue, right? And one of the key reasons for that is coming back to this acetyl-CoA. So here's the breakdown of carbohydrates. So we see our glycogen, glucose, right? Here's the process of glycolysis. There's our pyruvic acid, acetyl-CoA into the Krebs cycle. Here it is for the fats, triglycerides, right? What it's not doing a great job of showing is that glycerol can go this way and fatty acids come down here. It's trying to just point to this term. But anyway, with both cases, you get acetyl-CoA. And if you are consuming too much carbohydrate, right, we can easily go from this acetyl-CoA upstream to our glycerol and fatty acids and then convert that via lipogenesis to these stored fats in the body. Acetyl-CoA is the starting point for fatty acid synthesis. So let me kind of walk you through. So if we've consumed too many carbohydrates, and we have capped out our stores of glycogen, which doesn't take that much, right? Excess glucose in the bloodstream. We know we go through glycogenesis to create that glycogen. We store it up in the liver. We store it up in the skeletal muscles. But those stores have limited capacity. So once we're finished storing those carbohydrates, guess what? We make fats out of them, and they go up here. And again, it's because this acetyl-CoA is this intermediate. This can be a useful graphic to try to explain all these different processes in your mind. So all that's being shown here is hours of starvation. So there is actually a difference between starvation and fasting. So starvation is unintentional, where the person doesn't have a choice, perhaps in malnourished countries. Whereas fasting is intentional. And we're seeing that with intermittent fasting, more and more people are fasting for 12 to 24 hours. But this is going to be, we're looking at some of the hormones and some of the metabolites that you would see change over the course of these 24 hours. And this should start to make sense now. So insulin, right? We see that insulin rises when blood glucose is too high. So it's no surprise that as this fast continues, insulin will drop. We no longer need to store the glucose, and instead, we need to break down the glycogen to get the glucose. So you see the glucagon, the hormone coming from our pancreas, rising as we undergo this fast. coming down here, blood glucose would begin to drop, right? And that's one of the reasons that you see that. So the body's trying to build that blood glucose back up. Free fatty acids. So as this fast continues, and if we're not getting enough glucose from the breakdown of glycogen, blood glucose will continue to drop. So what our body will do is it will break down more fats. It will do more lipolysis to free up glucose. So you start to see more free fatty acids. Liver glycogen, right? So think about that for a second, right? No surprise that that tanks, right? We use up all that glycogen in the process of glycogenolysis, right? We split it to free up the glucose. But again, we run out of that. We run out of that source. And so we start having to depend more on fats. And so the piece of this that I haven't yet addressed are the ketones. So ketogenic diet is huge right now. And I want to try to explain a little bit about why those ketones are produced and where they come from. Okay, so you've got just this big overview of lipid metabolism here. So just to review, so here are our triglycerides. If we undergo lipolysis, we're splitting it, glycerol, fatty acids. So we've seen that. Glycerol is treated like a carbohydrate. You get your pyruvic acid, etc. Fatty acids, meanwhile, go straight to the acetyl-CoA via beta oxidation. Okay. And so if your carbohydrate intake is inadequate, intentionally or not, lipolysis, so this whole process speeds up to try to provide the body with the fuel, with the glucose that it's not otherwise getting. And in doing so, what you see happen is acetyl-CoA builds up. It's one of the reasons that it builds up is there's an important intermediate here. So you have oxaloacetic acid that brings in acetyl-CoA into the Krebs cycle. Well, that's actually used up. Just again, side note, you don't need to know this for any of my tests, but that oxaloacetic acid is used up in gluconeogenesis. And so it's not there to be able to bring this acetyl-CoA into the Krebs cycle. So the acetyl-CoA backs up. It starts to build because it can't get into the Krebs cycle. And look what happens. Look what the body does. So we undergo ketogenesis in the liver, and we get what are called ketone bodies. Now, I've really emphasized that glucose is needed by the central nervous system. The brain can also use ketone bodies for fuel. So this is, a mechanism that has evolved in the body to protect ourselves in times when we have to go for extended periods without enough food. Last up, protein metabolism. So as I alluded to before, with the proteins, we really don't like to use proteins if we don't have to for fuel. And I'll explain why in a moment. So we are really emphasizing the anabolic reactions in the case of proteins. So proteins from our diet, eggs, meats, dairy, yogurt, what have you, peas, we break down those proteins, we get the amino acids, and then our body immediately takes those amino acids and converts it to whatever proteins we need. And just the proteins in the body are countless. Every enzyme is a protein. Think about all the proteins on the surface of all of our cells, the collagen under our skin itself, right, has a lot of proteins in it. Our muscles, smooth muscle, skeletal muscle, cardiac muscle, right? So these are critical building blocks to the body. And so we're not going to break those down if we don't have to. And we're going to use those instead for anabolic needs to make protein. Now I have this, the all or none rule. So what this says is that, let's say you need 15 specific amino acids to make a protein. And if you have 14 out of those 15, you can't make that protein. And those amino acids will just go back to our amino acid pool and we'll use it to make a different protein. So we have to have all those amino acids present to make a protein because we don't store proteins. We don't store amino acids. And I'm trying to emphasize that because we do store extra carbs. We do store extra fat. Extra carbs goes to glycogen. When we run out of room with the glycogen, it goes to our adipose stores. Extra fat definitely goes into adipose stores, right? But if we stored extra protein, right, extra amino acids, right, that's acidic. That would change our pH. So there's no place in the body for us to store those proteins. We make, check out this stat, 500 to 1,000 pounds of protein over the course of our lives. It's amazing to think about. So if protein available from our diet is more than what's needed, is more than what's actually needed to make more proteins, then we will convert those extra amino acids to fat. We'll actually can undergo lipogenesis here for storage, or we are forced to break down those proteins for energy. And so let me talk about that a little bit. One of the reasons we don't like breaking down proteins for energy is because it makes a toxic waste product. Lots of details, many of which you don't need to know, right? So take home points here. So if we have amino acids and we need to break those down, right? Oxidize, breaking them down for energy. One of the reasons we don't like to do that is we have to do a lot of extra steps with carbohydrate breakdown, with fat breakdown. It's very straightforward. Protein breakdown, not as much. We have to first kind of convert. It's almost like we trick the body and try to convert the amino acids into a carbohydrate. And take home point, one of the byproducts of breaking down proteins is that you get ammonia, NH3. Ammonia is toxic. And our liver is going to help us combine that ammonia with CO2 to make urea. And then from there, our kidneys are going to be responsible for regulating the amount of urea through the urea cycle and ensuring that we excrete as much of it or excrete much of it in the urine. So we are immediately going to have to handle that ammonia. It's toxic, it's harmful. So the liver is going to say, nope, can't handle that. Let's make it into urea instead, and then the kidneys will deal with it. And so you get this byproduct. So it's a lot more work for the liver, a lot more work for the kidneys to be able to handle these excess proteins. So that's all I have on this chapter on metabolism, this focus piece on metabolism. So hopefully in reviewing these learning objectives, you can now distinguish among these, right? Try to turn to somebody else now and teach them. Teach yourself, teach your wall, teach your computer. And then can you distinguish among lipolysis, breakdown of fats, creation of fats, and ketogenesis? When would each of these occur? Are they going to occur during times of feast or times of famine? And same thing with the carbohydrates.